everyone, so today we'll be discussing how proteomics can enhance our understanding of targets in protein misfolding and cancer therapy. So for some background knowledge, cellular function is due to proper protein folding, and it is a crucial factor in a variety of diseases such as cancer. So proteins can misfold due to factors like cytotoxic stresses, lack of nutrients, exposure to amino acid analogs, etc. However, there are molecular chaperones that help with misfolding. For example, heat shock proteins like HSP70 and HSP90 can help cancer cells survive stressful conditions. However, overexpression of these is linked to chemotherapy resistance. So figure 1 shows a visual model of the 4-step process that researchers use to categorize protein misfolding and therapeutic targets in cancer. So in step 1, proteins are synthesized, folded, and transported to the Golgi complex and extracellular space. In step 2, misfolded proteins form prefibrillar oligomers, which can trigger apoptosis. These aggregated proteins are then targeted for degradation via macroautophagy. And then step 3 shows the therapeutic interventions such as curcumin, which can prevent misfolding, and HSP inhibitors such as HSP90 and HSP70, which block protein aggregation and also help with degradation. The last step is mTOR inhibition and clearance, where the mTOR inhibitors like rapamycin, CC1-779, RAD001, and AP23573 increase macroautophagy to help clear misfolded proteins. This inhibition of mTOR stops aggregates from accumulating and reduces the risk of apoptosis. Moving on, we have some oncogenes and tumor suppressor proteins in misfolding. So, for example, the C-terminus of the HSP70 interacting protein, also known as CHIP, helps degrade misfolded proteins and suppress tumorigenesis. mTOR signaling can also be disturbed by misfolding and help progress cancer. Finally, the proteomic strategies to help further understand the topics discussed and target misfolding involve tools like 2D gel electrophoresis and mass spectrometry. We also have curcumin, which again can prevent protein misfolding and aggregation, and rapamycin and its analogs CC1-779 and RAD001, which are mTOR inhibitors that could help fight protein misfolding related to cancer progression. This topic should be included in the BCH 261 course because it is not only interesting to learn about, but it exposes students to specific real-world techniques to help them understand the targets in protein misfolding and how it applies to cancer therapy, which is a common health issue in the present day. It also reinforces the understanding of protein folding, misfolding, and disease, which is already a topic in Chapter 6. So proteomics would give a deeper applied understanding and allow students how to learn how proteomic tools are used, which can then set the foundation for future findings of therapeutic targets.